Good morning, everyone. I'm Solène Brohard, and I'm part of the CNRGH, the National Center of Research on Human Genomics in the Laboratory of Bioanalysis. I present my poster on the study of high-order chromatin interactions in regulatory regions with the Pore-C approach. Pore-C technology is quite similar to IC, but the use of nanopore technology enables read sequencing containing more than 2 fragments in contact without amplification. From sequenced reads, we applied the bioinformatics pipeline that is the first version proposed by the Chmielynski laboratory. This workflow is based on genome local mapping to recover the genomic regions present in the nanopore read and on graphed approaches to generate the concatemers. A concatemer is composed of the defined genomic regions implicated in the interaction. Its cardinality, also called concatemer order, is the number of fragments containing each concatemer. From the multi-array interactions, the workflow also generates virtual pairwise interactions to compare Pore-C with IC data. We launched this pipeline on each Pore-C dataset, the 2 public datasets from Chmielynski lab, and the homemade ones obtained from the same lymphoblastoid cell line. We first studied the concatemer orders. We wanted to compare the proportion of concatemer orders in each sample. We observed that using NLA3 at the digestion step generated concatemers of higher orders. This more frequent culture enzyme allows studying multi-array interactions implicating more than 20 regions. On the other hand, we validated the Pore-C data by comparing the virtual pairwise with IC data on the same cell line and using the same restriction enzyme DPN2. The number of pairwise interactions in Pore-C is lower than IC. In the contact map of this chromosome 3 region, used as an example, we observed that despite the lower depths of Pore-C, we could detect loops, but also high-order chromatin structures like topological associated domain. To analyze this multi-way interaction, Deshpande et al. Proposes an R package called Chromunity. The tool first divides the whole genome into bins of a specific size. Only the bins intersecting regulatory regions of interest are retained for further analysis. Then, based on half of the sample interactions, it will research bins that frequently belong to the same concatemer named as a bin set. A list of bin sets that frequently interact are generated by sample. From a background probability model, it estimates an expected value of counts for every bin set. For each of them, it computes all the bin combinations of all possible order sizes. It calculates the excess of observed counts covering each bin combination compared to the expectation. For each bin set, it tests if there is a significant enrichment of higher-order contacts compared to pairwise. From our local data and only on chromosome 3 containing around 27 million interactions, we tested the computational limits of the package. We observed that no chromunity could be detected with only 1 million interactions, but with the recommended use of the package, we couldn't analyze more than 10 million contacts due to excess memory. We decided to randomly separate chromosome 3 interactions into 3 subsets of 9 million contacts. We compared the regions belonging to bin sets enriched in higher-order contacts found by chromunity. Unfortunately, very few regions are commonly found between the 3 interaction subsets. We also compared how these common regions are distributed into bin sets, but they did not appear to be part of the same interaction hub. The computational limits of the chromunity package made it impossible to obtain robust and reproducible results. We now plan to increase the computing capacity to explore at least the whole chromosome, and we hope to detect significant interaction here. Thank you for your attention.